Hello and welcome. This webcast is part of our series on antimicrobial peptides. I'm Alistair Freeland, and with me here is Dr. Xavier Just from the Institute for Research in Biomedicine in Barcelona. Dr. Just, many thanks to you for taking the time to speak to us today.
Well, thanks for having me, Alistair.
Dr. Just, for research into tyrosine A analogs, this research shows us that you can deactivate antibiotics as soon as specific wavelengths are present. Could you guide us through the research process, maybe, and what challenges you encountered and how you tackled them?
Yeah, so, well, this project arose from, basically two ideas. So my main motivation was here to design new kinds of antibiotics that change a bit the paradigm on how we think about them, not just something that can kill the bugs and remain active afterwards, but also compounds that we could deactivate so they stop killing the microorganisms once they are released into the environment. And quickly using light for that became like a very obvious option, because in these cases you can have a very clean stimulus, so because light won't produce any waste. And of course, you don't want to use any kind of light. So using visible light that was safe for healthy tissue, because in most cases UV light is used, and that can be a cause for cancer in living tissue and can produce mutations that are obviously undesired. So there have been some recent reports on the use of visible light to change the conformation of molecules, what we call photo switches. So it's like a molecular switch, something you can change the shape of by using light. And these new ones using visible light have become available quite recently. So the idea was to merge both things. So using visible light that was safe and also deactivate the compound afterwards to reduce the chances of antibacterial resistance appearing.
And your work shows how antibiotics could be deactivated as soon as it comes into contact with natural sunlight. Because a lot of antibiotics end up in the environment, and antibiotic resistance is on the rise. Do you think such a kill switch or deactivation switch could be put on for all antibiotics? Or do you think new antibiotics would have to be designed around such a switch?
Well, I think that both things can be done, and that depends on what the parent drug is. So obviously there are some drugs that are already available that will have a handle where you can attach this photo switch. And if you're lucky, you might be able to regulate their activity by switching conformations with a light source. But that will not always be the case either, because you won't be able to selectively attach that photo switch to one specific position, or maybe the position where it doesn't modify these antibacterial properties of the compound. But in those cases, you can always go around this and maybe start from scratch. So synthesize the drug around the photo switch. And both strategies have been used. I think that depends on each case. We have actually published results where we synthesize de novo the antibacterial peptide instead of using the natural product that is already available. But we also have programs going on where we just take available drugs and functionalize them. But that's work in progress.
And do you think bacteria could gain resistance against such antibiotics, or maybe the inverse? How could resistance against such antibiotics come about?
Yeah, well, it's not impossible, of course. The thing is that these switches, and of course the molecules attached to them, are not always present in just one form. So they don't only exist in either conformation. You always have mixtures of different ratios of the two shapes of the molecule, let's say. So there is a lot of work trying to address this and getting like nearly to 0.5% of one of these isomers that we call them. But obviously, at the moment, that's not always available, and it's not always possible, especially if you want to use visible light. So at the moment, you will always have a little amount of the undesired isomer of the drug. So even if it deactivates with sunlight, you will always have a tiny amount of the active drug. And that, of course, that will be present at a very small concentration. It might not kill bacteria, or will do so in a very large, a very small scale. And that means that there's always a chance for some resistances appearing. But in any case, the rate at which that will happen will be very slow, much slower than the actual rate.
And in what way could this concept be, do you think, improved on? Do you foresee more antibiotics with a deactivation switch, or where do you see us going?
Yeah, well, I think that if this is not the way to go, at least it's a very promising one, because of course there are many other approaches that you could think of. There are very few but interesting reports introducing other ways of deactivating the drug. For example, there was a report last year where the compound is actually degraded by enzymes that are present in soil microorganisms. So it will survive in your body as an active compound, but once released, it will be quickly degraded by bacteria in soil. So it won't exert any evolutionary pressure on those other microorganisms. And with light, we can achieve something very similar, at least with the system we're using for visible light. And we still have to see how far it goes. But in any case, I think that innovative approaches that, as I said before, escape from just having a drug that remains in the active form forever and accumulates in the environment is something that, I mean, we have to change for sure, because that's leading to the present problem of resistances being quickly spread.
But do you still see any downsides in such a deactivation switch? Yeah,
I mean, the current state of the art is not perfect. I mean, it's a very young field, because the first report for the switch of antibiotics was published in 2013, if I recall properly. So it's a very young topic. The first report using visible light was only 4 years ago. It was published in late 2017. So we actually started working on this soon before this first report was published. And there's only a handful of results on this area. So sure, there's a long way to go. And I hope we can improve these ratios of isomers that I mentioned before. And we can come up with a much broader variety of compounds that show activity. And of course, it's always desirable to improve the selectivity against certain strains. Because if you have what is called a broad spectrum antibiotic, even if the compound is deactivated afterwards, or it's only a tiny amount of the compound that is active, you will always have chances of killing bacteria that you didn't want to kill in the first place, and that will might cause resistance in those other microorganisms. So ideally, we have to go for antibiotics we can deactivate, but that once they are activated, they only kill the one bug you want to kill, but not the others. I think those are all the requirements we need to address in the future.
And maybe one more question. Is this something you are working on at the moment, or could you share with us a bit more what your current project is that you're working on?
Yeah, so we actually achieved some of these requirements that I mentioned with the first publication. So we can use visible light, and the compounds get deactivated with only sunlight or even with ambient light from a white bulb. And we also identify a couple of compounds that were quite selective for one specific target. at least among the ones we tested, which was a decent size collection of strains. And we are pursuing these compounds further. We want to test them against a broader collection of bacteria. And we also go to higher potencies for this compound and get also better activation rates. So at the moment, we can have this activation with light, but the potency of the compounds is not something amazing. But we can see that the non-active compound is fully inactive, and the activated ones are active to a certain extent, but we have to improve on that. And we also want to achieve selectivity against other more relevant, perhaps, strains of bacteria that are the ones causing trouble in hospitals, for example.
Dr. Just, many thanks for taking the time today to speak on this subject. Good luck to you and your team for the further work. And I hope to see you again at some point in the near future. Thank you.
Thank you very much, Alistair.